Okay, guys, so I just counted how many people were in class. And we are over half, but not by much. So I guess that's a bad thing about having exams and recitations, that people will not come to class.
How many people have a recitation this weekend? Yeah, you should call us. Yeah, you should. Yes, your Wednesday recitation.
Who's in Wednesday's recitation? Pretty good. Well, then the other thing is usually as the semester goes on, people start to think, oh, I don't need to come to class because she recorded that here. So that could also be a problem, which is all fine with me. Okay, so let's move on. So the first, what's my topic? Oh, the little ball. My son wanted to bring this to school. He's 2. Right? Not good. Esophagus size. And so his teacher was like, give it to mommy. Thank God his teacher said it, because if I had said, give it to me, he would have been like, no! And then had a fit. It's all in my pocket. Okay.
Don't lose it, you're in trouble.
I know. Well, just forget about it. Their memories are horrible.
Girls, yeah, no, boys, yes.
Okay, so the first the first cohort took their exam on Monday, and I mean I haven't graded it yet. Do you think I'm crazy? I'm working on it though. It actually is nicer for me because it allows me to get a head start on the grading in a weird way. But I feel like it was good because most people finished within the first hour, and some took till the last minute, but there's always going to be those people who want to hang out with me until the end, until the bitter end. Do you have a question?
Is there any way people in Monday recitation
No one's grades will be posted until they're all graded, because that could create a fair amount of anxiety in those who haven't taken it yet. So there's no need. Fair amount of additional anxiety. Okay, so let's learn about cell cycle. Who loves the cell cycle? Oh, good. Eat some class. Oh wait, are you guys dying to ask me how my grant submission went?
Yeah.
Thank you. Horribly, actually. So here's a life lesson. You know when you guys want extra time for things, and a professor says, no, this was the deadline, you lost it. So NIH, the way it works is your grant needs to be in by 4:59 and 59 seconds or else. That's what they tell you. And despite everything being ready on Saturday, certain things, Hopkins, slow things down. And whatever. So when you submit a grant, you put it in the electronic system, and then the computer goes through to make sure there's no errors. If there are errors, it kicks it back to you. You have to submit it again until there are no errors. And so we had to submit it 4 times, and the timestamp was 5:11. It did not get automatically kicked out, which is weird, which means they're big liars maybe, I don't know. So I do not know if it went in or not, but it's a huge lesson. I was talking to Dr. Beck, and he was like, it's just like you tell your students, there's a deadline, you need to meet it. And I was like, shut up. Sometimes there are things that happen, like whatever. So who knows what happened, but it was slightly stressful because I'm like sitting there and I'm getting these emails that are like, action required, your submission is not complete. Go back in, fix it. Anyway, so we'll see, because they should have the opportunity to see my brilliance. And maybe they will, maybe they won't. Okay, thank you for asking. You guys are also thoughtful. Okay, so last time, so the lesson for you guys is be early. Don't be on time, be early, because you never know when technical difficulties are going to get in the way. And though I might be understanding of technical difficulties, large institutions like the NIH are not. Their late policy clearly says, we don't care if there's some issue with errors, everyone has that problem. When's the next submission? June. Okay, so last time, what did we talk about? We're talking about the cell cycle. What is it? You guys all know. And we were talking about how we study the cell cycle. We spoke about two model organisms. The one that we will look at the most in this class is yeast. We talked about how you can study different regulators of the cell cycle by visualizing mutant phenotypes. Again, looking at yeast that don't divide properly and complementation assays. We talked about also just visualization of cells under the microscope, got our stain with antibodies. We talked about BRDU incorporation. We talked about flow cytometry, and that's where we ended, and that's where we will start today. So this is where we ended. I was talking about flow cytometry, and I showed you this picture that I stole from the internet. Where basically it's just showing that you've got your cells dripping through, and then there are lasers that shine, and depending on how the lasers bounce off the cells, they're detected, go into the computer, and it counts each cell and can group them based on whatever you want. It could be forward scatter and side scatter, so how the light scatters in different directions. It could be based on an antibody that you stained it with. If it's green, how many cells are green, stuff like that. So this is an example of how you could use flow cytometry to measure whole blood. If I take your blood and I spin it down, so I get rid of all your red blood cells, because those are trouble. Not really trouble, but maybe I don't care about them. And I'm looking at your all your other white blood cells. Now you know there are a lot of types of cells in the blood other than just red and white, right? So there's a lot of white blood cells, and you can see the way, so this is just forward scatter versus side scatter, and it's plotted on this graph based on where things, so each individual dot is a cell. And so this is side scatter and this is forward scatter. And it's just how the laser that's being pointed at the cell, how the light scatters and how the detector picks it up. And you can see that you get clear populations of cells that have similar profiles in terms of how the light scatters. And this is routinely done when people are looking at whole blood. I've looked at a lot like in clinical studies where people are taking blood out of patients and trying to see immune responses to things, because certain cells will go up, certain cells will go down, especially in HIV research. But here you've got these cells are granulocytes, these cells are lymphocytes, these cells are monocytes, and just for you to understand that this is how powerful flow cytometry is. It's even more powerful than that. Oops. You can analyze these same populations of cells with antibodies, right? You can treat those cells with CD45 or CD3 or multiple things. This is side scatter and CD45, so you get your population of cells that have that same light scattering ability, so the same shaped cells, and then you can stain them with CD45. So these are your populations of cells. They're all up and down here. And then here's CD45. And the greater the intensity of your staining, the more to the right you'll go. So these cells are all CD45. Don't worry about what CD45 is. This is not an immunology class, but it's a cell surface marker that's frequently used to distinguish different populations of cells. Here's another example of the way you could do it, where you'll stain with two antibodies, one against CD4 and one against CD3. These are going to get your T cells. Specifically, there's CD4 and CD8 T cells, so these will just get your CD4 T cells. And you can see as you move outwards, your CD3 staining increases, and as you move upwards, your CD4 staining increases. So the ones that are out here are CD3 and CD4 positive. Here's another one where they're looking at CD8 and CD3, and it looks, as you move up, it's your CD8 positive. As you move out, it's your CD3 positive. So this population is your, what are they saying, CD? Should be CD8. And then here's another one where they did CD8 and CD4. And so what that does is your CD8 are going to end up here, and your CD4 are going to end up over here. And this big blue blotch over here are ones that are neither CD4 nor CD8. Because remember, in your blood, they're not all T cells. There are other cells there. And then here, they're stating another thing, TCR alpha beta, which is the T cell receptor. Here they're looking at all T cells. So this is TCR, and then CD3, which is, again, all T cells. And you're getting up in this population here, these are TCR, CD3 positive, TCR cells. Yes?
When I'm testing for CD4 positive and CD3 positive, why do I need to use CD3 as well? So I can just test for each.
So when they're testing for CD4 and CD8, why are they doing CD3 also? Well, CD3 is a way to get all of your T cells, and CD4 is a way to get a subset of your T cells. So it's a more stringent way of isolating CD4 or CD8 positive T cells.
So T cells are not ones that contain CD3.
T cells always contain CD3. Though this is not important for this class. It's just an example of how. So do not get bogged down with CD4. This is irrelevant to molecular biology. It's just an example of how flow cytometry works. And it's fairly powerful because you can stain for anything. Any other questions? Okay, so you guys need to memorize all the CD antigens. I think there's like 200 or something like that for tomorrow. Okay, just kidding. Okay, so why am I talking about this? Because you can evaluate cell cycle through flow cytometry by quantifying DNA content. And this is through staining with propidium iodide. So to understand this, you need to understand that your cellular DNA is going to change in terms of how much is in each cell through the cell cycle, right? Right? How does the DNA change in terms of quantity during your cell cycle? Oh wait, let me get prepared to call someone random. Ryan McGee. Sorry if I've already. Where are you? Okay. You had a big skull and crossbones next to your name. Okay. I feel like I've been really bad at crossing people's names off, so I'm sorry. Kelsey Winters. Everyone's sick today, huh?
No, they're in the Sarah's in Wednesday recitation.
Huh?
Kelsey's in our Wednesday recitation. And so is Ryan.
Okay. Where is Amanda Talbot? Oh my gosh. They're all going to hear this when they listen to the lecture. That'll be fun. Okay. How about Charles Hawkins? Chip. The question was, how does your cellular DNA content change through cell cycle? It doubles in S phase, or technically at the end of S phase. Right? So it increases through S phase because you're duplicating your chromosomes. Absolutely. And that's what the PI staining is based on. Okay? So you can stain your DNA with this DNA stain, just like DAPI, but it's PI. So DAPI's blue, PI's orange, and the cells with the highest amount of fluorescence have the most DNA. So what phase of the cell cycle do you think you will have the highest fluorescence or the highest amount of DNA? Where's Yun Lee? Yun? Yeah. Oh God, this is sucking. All the people who are panicking. Inango. So the question was, what phase of the cell cycle will you have the most DNA? Think a little bit more. S phase is when you're copying your DNA. You can stick to more broad phases. Okay, let's try again. Who wants to answer this question? Yeah.
The G2 and
G2M, right? Because you finish replicating your DNA. Thank you for playing. You finish replicating your DNA at the end of S phase, but you don't segregate and become two separate cells until the end of mitosis, right? So you're going to have double the DNA content until mitosis is done, and you go through cytokinesis, right? Who's confused by this conversation? Okay.
What was the question you were asking?
I'm not telling you. Who said
I
that?
think I'm answering the wrong question.
Okay, so what phase of the cell cycle do you have the most DNA? Okay, so remember you've got G1S, G2N, right? So you have many chromosomes. You've got, let's do per one chromosome, because you only have one chromosome. S phase 2, right? You still have 2, right? You still have 2 until cytokinesis, right? And then you go back to 1. Am I right? Am I right? Does anyone want to argue with me?
So
Yes.
why is an S phase included?
Because that's not your most. That's not the most. The question was, which phase of the cell cycle has the most DNA? S phase, you start with 1, and throughout you increase.
There you go.
So it would be everything from G phase to cytokinesis?
Everything from the end of S phase through cytokinesis. So let's look at this. Let's look at this graph here. Okay, so this is what this is showing. You stain your cells with an orange stain called propydiamidite, and it stains all the DNA in your cells. And you have, this is intensity of staining. So the closer you are to 0, the less orange. The further you are from 0, the more orange, and that correlates, right? If you have more DNA, you're going to have more intense staining, right? So before I showed you dots everywhere on a graph, this is just showing it plotted differently. And so you'll have a number of cells, you'll have, 70% of your cells will have this much intensity. 10% of your cells will have this much intensity, and maybe 20% of your cells will have this much intensity. So this is a very low intensity of staining, so it's less DNA. This is twice as intense, so it's twice as much DNA. Okay? And so you can ask a lot of questions about cell cycle, or how different things affect cell cycle. And then look at this, because this is going to shift. Let's say you have some drug you're treating with, and it prevents you from exiting mitosis. Well, so this is going to go away, and all the cells are going to move over here, because they're all going to go through S phase into M phase, but you can't exit mitosis. So this will be low, and this will be a really high peak, because remember, this is your number of cells right here. Okay? Yes?
Why does the number of cells change?
You're counting, let's say you're counting 1,000 cells, and it distributes those 1,000 cells along this graph.
Right.
So it's the proportion of cells.
Okay, I see.
Okay? Yes?
what's going on with the cells that are below 1 and above 2?
What's going on with the cells that are below 1 and above 2? So if they're over here, they're apoptotic or necrotic. If they're over here, I'm guessing they're polyploid. I don't know. Some sort of strange, much more than 2 copies of your chromosomes. But you're not, so if you're just talking about this tail, that's part of that too. So you're taking into account your peaks. Yeah. Any other questions about this? So those of you who had me for cell saw this before and hopefully understand it. Yes.
With those tails, that's what the 2 sided tail have to do with the numbers.
Don't even go that far. You're gonna be in two different peaks, okay? And the peaks are gonna be 1 copy of DNA and 2 copy of DNA, and that's it. That's it. Don't go any deeper into it. This is a well-established method, and even if you have like huge amount of telomerase activity, you're not gonna triple your DNA content. But this is about DNA content per cell. Any other questions? Does everyone love this? I think it's actually really cool. Okay, so let me just show you another picture. So this is another way of looking at it. This is PI staining over here. So intensity of PI staining, it's just plotted in dots rather than in a graph. Well, this is a scatter plot versus a histogram. And your PI staining intensity is increasing. So over here, these have low PI staining, and over here, these have high PI staining, and in the middle, it's somewhere in the middle. So this would be probably G1, this would be S, and this would be G2M over here. And what they did is they chose another stain, which was for phosphohistone H3. I did not tell you this, but phosphorylation of histone H3 correlates with chromosome condensation during mitosis and meiosis. So it's a good marker for mitotic cells. And so the cells that have high staining intensity are up here. And so the ones with high staining intensity here and high staining intensity here are right over there. And those are your mitotic cells. So they have high PI staining and phosphohistone H3. I was actually looking for some like acetylation, methylation related ones to show you, but it wasn't this, it didn't work the same because whatever, this was prettier. And so this is, if you do immunofluorescence and you stain with perpidium iodide, this is, remember I told you the PI is orange and it stains the DNA. And so those are your nuclei. And then the green is just antitubulin. You see your microtubules. What phase of the cell cycle are these cells in? Antwi. I saw you. Oh, there you are. Interphase? Yeah, absolutely. Interphase, right? You do not see any condensed chromosomes anywhere. And your microtubules are in this kind of interphase organization versus forming a mitotic spindle, right? Any questions before I move on from here? Okay, so hopefully in the next 3 years I'll be doing some of this in my lab if I get my stupid grant. So you guys are welcome to come watch. You know, those of you guys who are still here in 3 years. Who's going to be here in 3 years? You guys are all going to be moving on to bigger and better, right? You're going to be like, oh, Towson. It's glory days. Okay, so we're going to spend a little bit of time talking about cell cycle regulation. Not too much because I mostly do that in cell biology. And then we'll go into S phase to talk about DNA replication and then into mitosis. Okay, so cell cycle regulation by and large depends on the activity of cyclin-dependent kinases, CDKs. What's a kinase? What does it do? Oh, Rosalind Melgar. It phosphorylates. They phosphorylate a few different things. One is tyrosine. Depends on the kinase. Kinases are sometimes fairly specific. But yes, phosphorylates. And what does phosphorylation tend to do? Gregory Irwin. Is Mary calling today? Oh, there you are. Changes shape, right. So that's how you'll get regulation of things, because if you phosphorylate something, it'll either allow something else to bind and do something, or might prevent something else from binding and doing something. So, so these CDKs are the key to cell cycle regulation, and CDK activity is controlled by binding to cyclins. So when a cyclin binds to a CDK, it's active. If no cyclin is available to bind to CDK, it's inactive. Any questions about this concept? It's a major concept of cell cycle. Okay, good. So how does the cell use regulation of CDKs by cyclins to control the cell cycle? Well, basically what happens is you get this up and down of cyclins. You get increased transcription, translation of cyclins as the phase of the cell cycle that is going to start approaches, and then at the end of that phase of the cell cycle, you'll get degradation of those cyclins. And so as you get increased transcription, translation, the proteins build up, the cyclins become available to bind the CDKs and activate them, and when it's time for that cycle of the cell to end, then the cyclins are degraded. And so you can see here, G1S comes up in G1, and then it's around to help start S, and then it gets degraded. S cyclins come up, that's what really starts S, and it makes it all the way through here, then it gets degraded. M cyclins come up right as M is starting, and then gets degraded. Actually, M cyclins need to be degraded to go from metaphase to anaphase. So it's this rise and fall of the cyclins that determines what phase of the cell cycle you're going to be in. Yes?
So each one of those phases have their own cycles,
Yes.
and they're different from the other ones.
Yes. So that you can think of it as cyclin CDK complexes. Every CDK has its own cyclin, and each phase of the cell cycle has its own cyclin CDK complexes. So the CDK might be around in a different phase of the cell cycle, but it won't be active because the cyclin won't be there. How are we doing? This is fun. Cell cycle is probably my favorite, I guess. I don't know. Transcription's good too. Okay. So cyclins rise and fall through the cell cycle and activate CDKs and then directs them to the correct target proteins. And whatever target protein is phosphorylated, some kind of process involved with that phase of the cell cycle will be activated. Okay, so this kind of relates to the last comment. Here's your picture of the cell cycle over here, and you're going to have different CDKs and cyclins activated in different phases of the cell cycle. Okay, so here's an example of what's happening during cell cycle regulation. Now, this is a figure out of the old textbook that I used last year, and I kept this one because it shows the intricacy. Well, it shows a very not intricate version of the intricacies of cell cycle because it looks really complicated, but it's way more complicated than this in reality. So you'll have your S cyclin CDK become activated before S phase, right? And that in turn will prevent M cyclin from being degraded. And so that allows M cyclins to start to build up because they're going to be needed over here. And that will so then the M CDK will get activated because your M cyclin's built up, and that will turn on right over here. But that will also prevent S cyclins from functioning because that activates degradation machinery here that will then degrade S cyclins. So there's this huge network of this rising and falling and making sure the cyclins that don't need to be active are not active, and making sure the cyclins that do need to be active are active at the appropriate time. And when this becomes dysregulated, it's a problem. Okay, so there are many levels of CDK regulation, and this is what assures tight regulation of the cell cycle. Active CDKs are required for phosphorylation of proteins, required for different phases of the cell cycle. So what you always need to be keeping in mind is how are these CDKs being regulated, and what are the mechanisms of regulation? And we're going to go through that shortly. Questions? Comments? Concerns? Fears? Things you want to share with me? Okay. Is anyone asleep? No. I used to have a guy who would always fall asleep. Right in the middle. He'd sit like right there. Right there. Did you have a question?
No, I'm just tired.
Oh, okay. You're tired. I hear you. Yes.
Is the inactivation of metaphases and
Is the inactivation of metaphase, anaphase, checkpoint, is that what leads to aneuploidy? Well, the metaphase anaphase checkpoint is activated by incorrect spindle attachment to chromosomes. So probably is involved in incorrect chromosome segregation, which would result in aneuploidy. That's logical line of reasoning. Yep. I don't know exact mechanisms off the top of my head, but I bet if we looked it up, we would find something about that. Okay. Any other questions? Okay, so CDKs are activated by cyclin binding, which is what I already told you. And what happens when cyclins bind to CDKs is there is a conformational change, which results in it results in exposure of a site that needs to be phosphorylated by a kinase called CAK, CDK activating kinase. So here's your inactive CDK, and you can see this red thing here as part of the structure, which they're highlighting. Cyclin comes and binds, and that results in a conformational change, which allows this loop to come out. And then CAK comes along and phosphorylates that, and that fully activates the cyclin CDK. So it makes sense. You wouldn't want to just have one level of regulation where cyclin levels can go up and you activate. You would probably also want to control your levels of CAK2 because that phosphorylation is going to be required. Yes?
So, how is the cyclin binding to the CDK if it has that one?
How is the cyclin binding to the CDK if it has this here?
Does it just push it in?
So specifically, it's just that there's a binding site for the CDK with a cyclin, and that changes the conformation. So this might be required for the binding, and then once it binds, it changes the conformation, which causes this part to stick out. Okay? So just because something's sticking out doesn't mean you're not going to have interaction. Proteins aren't flat surfaces that interact with each other. There's frequently all this different topography that's required for interaction between proteins. Questions? Okay. So there's also a way to negatively regulate cyclins or cyclin CDK complexes, and this is through another phosphorylation, which is inhibitory. So you can have activating phosphorylation, and you can have inhibitory phosphorylation. So here's your CDK, and it's bound by your cyclin, and you've got your activating phosphate group there. You can have another kinase called WEE1 come along and phosphorylate it, actually right in the active site, and that will prevent the CDK from being able to phosphorylate it. And so that's a way to keep it off when it's not needed. So you might want to have a buildup of this like almost active cyclin CDK complex, and then keep it inhibited. And when it's time to activate it, you can then activate a phosphatase that will remove your phosphate group. So these are just many different ways of regulating the activity of these very important kinases. So here's another way to regulate. There are these CKIs, cyclin kinase inhibitors. They do exactly what they say they do. Cyclin kinase inhibitor, they inhibit. Yeah.
Can CDK be used like over and over and over again when you're like after it's just used twice, for example, if it's integrated?
CDKs, can they be used over and over and over again? As long as they're active, they will be active. Yeah, so they can phosphorylate many, many substrates until they're inactivated, either by removing and activating phosphate group, or by degrading the cyclin, or by increasing CKIs. Okay? So there are many, so to go further into your question, there are many substrates for these kinases. It's not just like one protein is phosphorylated. So you'll have your CDK bound to your cyclin and it's been activated. And then you can have these inhibitors that will come along and bind the complex and distort the active site and keep that cyclin CDK complex off until needed. And that's frequently done when cyclin is being upregulated as you prepare to enter a phase of the cell cycle. Then you want to prevent the activity before it's needed. So you'll also upregulate P27 or another CKI, and that'll bind to the cyclin CDK complex and keep it inactive until it needs to be activated. How's it going? Yes.
So you can either use another phosphate or you can have a P27 do the same thing.
Yes, but different things are done at different times. Yeah, so you can have inhibitory phosphorylation, you can have activating phosphorylation, and you can have these CKIs. And it's not one or the other, sometimes it's a combination of multiple things going on at the same time. But these are different mechanisms. Yes? What would it take to remove the CKI? What happens is actually the CKI is degraded when it's not needed anymore, and we'll talk about that shortly. And so that might have been a good segue. That's an awesome segue. Thank you. I did not plant that, but recently I've been having some really good segues. Now thinking about those segues, and those are weird, aren't they? They're just kind of weird. At Hopkins, they've got all the security is on segways running around campus. And I always think to myself, like, if there's someone running, I guess, you're like, how good are they at driving those segways that they're not going to get killed while trying to chase down someone? Anyways, so now I'm going to introduce the ubiquitin proteasome system because, well, A, I work on it, so I think it's important that everyone who comes through my classes leaves with an appreciation. And B, it's critical for regulating the cell cycle because it's the cyclical proteolysis that controls the cell cycle. Okay? So this is just a figure showing that there are 3 enzymes involved in ubiquitination reaction. You have your E1, and that activates, that is ATP-dependent activation of your ubiquitin, and then it gets transferred to the E2, which either transfers it to an E3 or just binds to an E3. And then the E3 is the most important, in my opinion, the most important enzyme in this pathway because it is the specificity factor. It is the one that binds to the protein that's going to be degraded. Because if you have unregulated degradation of proteins, you have a dead cell. You need this very specific targeting of proteins to be ubiquitinated and degraded. For example, with the CKIs, there's a ubiquitin ligase that specifically binds to CKIs and degrades CKIs, or polyubiquitinates CKIs. So the substrate then is ubiquitinated and is actually polyubiquitinated when we're talking about degradation of proteins, and then it's recognized by the proteasome and degraded into peptides. You do not need to know E1, E2, E3, just understand E3. Really understand the E3 as the specificity factor. If you do not have an E3, you cannot ubiquitinate anything really. But that's what regulates which proteins will be degraded. And it's really important that certain proteins are targeted to be degraded at certain times. So the polyubiquitinated proteins are recognized by the proteasome, which is right here, and then degraded. And so this is just showing that you also have the ubiquitins are actually removed before the polypeptide goes into the proteasome, and the proteasome is just full of a bunch of different proteases, which then degrades the protein. So I just want you guys to really understand. First of all, I wanted to wake you up, because usually seeing Sesame Street doesn't really happen normally in 409, but I thought we'd wake you up with Sesame Street. By the way, Sesame Street's kind of awesome, isn't it? Who's watched it recently? Okay, thank you. I'm just saying, and actually, I just recently went through a whole lot of Mr. Rogers because of my kids. And I did not appreciate Mr. Rogers for what he was at all when I was like 4. But now I really do. He talked about some very difficult topics, such as using the potty. Okay, because you will not, he even has a song that's like, I won't go down the drain. Like you're not gonna, that's like apparently a fear for little kids that they're gonna go down the drain. That's where you start. So it's no matter how dumb you feel, you've like grown in leaps and bounds. You can use the potty. You know you're not going to go down the drain because you're too big, right? When you were 2, you were worried about that. I'm just saying. Okay. So the reason I put this up is I want you guys to come out of this class knowing that the proteasome is not just a cellular trash can. People who do not appreciate the proteasome talk about it as this is where misfolded proteins go. No. Yes, that's one thing that happens. But without this very specific, targeted degradation of certain proteins at certain times, you would have no cell. You would have nothing. And we all have cancer. It'd all be bad. Many cellular processes are controlled by regulated protein degradation. Think about it. If you want to turn something off, get rid of it, right? If this kinase is super important for S phase, degrade your cyclin and turn it off when you need to go in down phase. Okay? So everyone appreciates that? At least remember that Dr. Ehrlich showed you Oscar DeGraff. She talked about the body. Yes?
So when the protein is degraded, the particles are recycled.
The ubiquitin,
No, the protein.
the protein, it's degraded into peptide fragments, and then amino acids can be recycled, but it's not like it becomes that same protein again. Yes?
Why isn't it just a separate trash can?
Why isn't it? Well, that just implies that there's no level of specificity, and it's just where mistakes go.
Oh, okay.
So I just want you to realize it's more. It is a cellular trash can. It is, but it's not just a cellular trash can.
Okay, alright.
It's a very important cellular. It's like there's a recycling bin. No, it's a very important cellular trash can. So it's not. I've sat like when I was in undergrad. I feel like my professors told me, oh well, here's how we fold proteins, and if there's a mistake, they go to the proteasome. Isn't that what you guys learned? How many people learned that? I mean, if you had me, clearly I drilled this into your memory. But I really want you to know, it's not just like, oops, cell made a mistake. It's like, no, we're very much on purpose moving our cell cycle forward.
Okay.
Okay. Okay, I'm done ranting. 5 minutes. So transcriptional regulation and cyclical proteolysis regulate the cell cycle. That is the key. When you need more of something because you're moving forward in the cell cycle, protein levels will go up. When you need to shut something off, protein levels will go down. What are some examples of the types of proteins that you might want to degrade? Garrett Shaw. Garrett? Garrett? Okay. I see no hands. Kelly Cusick.
Yes.
What's an example of a type of protein that you might want to degrade at some point in the cell cycle in order to shut something off or to turn something on? You can phone a friend.
Wait, ask the question one more time. I really didn't.
I know sometimes it's hard to focus when the spotlight's on you, right?
Maybe a little.
Yeah. Should we do downward dog? You just have to forget the other people there. I always forget that you guys are here while I'm talking to you.
How about I don't know if it's a protein. I was thinking of receptor.
Yeah. So think in terms of cell cycle.
Okay.
And we just talked about there are cyclins, and they activate CDKs only at a certain time. And there are CKIs that inhibit cyclin CDK complexes only at a certain time. And sometimes you need to turn things off or turn things on. So can you think of an example of a type of protein you might want to degrade so you can either turn something on or turn something off? Yeah, a lot of friends around there. Cyclin. And she gets $1 million. Yeah, cyclin. Actually, Who Wants to Be? Is it Who Wants to Be a Millionaire? Where you get to phone a friend? I had a friend who was on that, and she only got like $25,000.
Only?
But like that's actually really bad for that show. She was also on Jeopardy. I knew her during my PhD, and rather than doing her thesis work, she was always trying to get on game shows. She works for the FDA now, so I guess she did okay. She did a great job. This can be on YouTube. She's not listening to it. Anyways, good job. So when might you want to degrade? I'm boring people. When might you want to degrade your cyclins? Someone else can answer. You don't have to be one. Yeah.
When you change what part of the cell cycle.
Right. So when you're leaving one phase of the cell cycle and going into another, you're going to want to degrade your cyclin because you need to shut off that CDK. Cancer happens when you don't degrade things sometimes,
You still want to.
but you want to. Yeah. You always want to degrade things if you're healthy. Okay. What types of genes might be upregulated at different phases of the cell? When I say upregulated, I mean transcription turns on, translation, more protein. What types of genes might be upregulated at different phases of the cell cycle? Jessica Mickey. Oh, yeah, I feel like I just called you. Cyclins again. Okay, right, because if you're going into, so this is the example, right? It's that increase and decrease that regulates the cell cycle. So cyclins again. CKIs are another example for both of those. You want to degrade. There's a lot of discussion. I'm so glad that I'm inspiring you guys to have a discussion over this. Thumbs up. CKIs will need to increase when they're needed, when they need to bind something and inhibit it, and they'll need to be degraded when you need to activate a cyclin CDK complex. Okay? How are we doing? I see one thumb up. 2 thumbs up. Okay? How about from the left side? Anyone happy? Okay, I got a thumbs up. Okay, well, we're pretty much done. I mean, I can teach you more, but you guys can go.